All right, folks, we're going to start lesson 7. We might have to finish it in our next class depending on time here. But this lesson is still about vision with a little bit more detail about how some of these things work. So we're just going to add a bit of information about photoreceptors. Now you'll recall that photoreceptors are located on the retina, but not on the blind spot. So there's a spot at the back of the eye where your optic nerve has to leave your eye, and there are no photoreceptors on that one particular spot. Now, if we consider the two types of photoreceptors that you have, one of them is called rods. Those are these ones here. They look kind of like a baseball bat, I think, when I look at them. Now, rods, as their function, detect light. And notice I really specifically did not include different colors. Rods detect light in black and white. So they're really detecting, is light present or is light not present? That's what a rod can do. So we're comparing it to the dark. Now, I don't know if any of you have noticed this, but when lighting conditions are really low, these are the type of photoreceptors that are functioning the most, and it's sometimes hard to see what color something is when it's really dark, and that's because a rod doesn't detect that. They are located mostly near the fovea, and the number of rods decrease as you get near the edges. So if I were to represent that on these little pictures that follow, if I'm looking at an eye, this picture is looking at an eye from the top, so I can see the front and the back. Where it says 0, that's your fovea. That's right in the middle of your eye. And if you look at the graph that follows, it's showing the angle relative to the fovea, which is 0, and how many photoreceptors there are. So if I follow the line for rods, it's this line. When we get to the blind spot, it drops down to 0 completely. Then it comes back up. Whoops, I started tracing the wrong line. And then it decreases as you get near the edges. So when I say that rods are mostly near the fovea, I mean that if I start at the fovea in the middle of the back of the eye and start moving towards the edges, this way or this way, the number of rods decreases. There's most of them in that middle part, and as we move towards the edges, the number decreases. Now, if we consider cones, cones detect colors. You actually have more than one type of cone. So there are 3 types that you have. We're not going to be specific about the 3 different types of cones. We're just going to talk about cones in general. But we have talked about people being colorblind before when we talked about genetics. When a person is colorblind, 1 or more of their cones is not doing its job, and the cone that's not working will determine which type of colorblindness a person has. The cones are these ones here. They look more like a cone, so they're pointier at the end than do the rods, and they're a little bit shorter. Now, the job of a cone, aside from detecting colors, is to really provide you with something called acuity, which we could reword as clarity. Visual acuity means how clear things that you are seeing appear. Now, there are other things that can play into visual acuity, but cones provide this. If we look at where cones are located, they are concentrated on the fovea. So if I trace the cone line, where are we here? It goes up really high right there in the middle. So they're concentrated on the fovea. Basically all of your cones are on the fovea, and very few or none of them are on the edges of your eye. So cones are right here. And so depending on where light is hitting your eye, different photoreceptors will be activated. If you're looking directly at something, it's mostly cones that you're using to see. But things that are coming into your eye from the side, like off in this direction for me, would be affecting mostly my rods because I don't really have cones anywhere other than the middle part of my eye. Now, does anyone want to ask anything about the difference between rods and cones? So for light to be able to hit your photoreceptors, something called accommodation has to happen. In this sense, when your eye is focusing on something, that's the process called accommodation. So you've maybe heard the word accommodation in other contexts before. This is a different use of the word accommodation. During accommodation, here's what happens. In your eye, you of course have a lens, and then you have that ciliary body and the ligaments that are attached to it that are attached to the lens. So if I look at these pictures over here, they're representing on the top just one side of the lens of your eye. There's a ciliary body and ligaments on both sides of the lens, but if we're looking at the lens of the eye from just one side, there's the lens, and here's the ciliary body and the ligaments up there. Now, if you know anything about how ligaments work, ligaments are part of movement. And when they pull or when they relax, like when a muscle pulls or relaxes, things move. So when they pull, that means they'll be tight. So if you look at the left side of my picture where the line is straight, that's trying to represent that the ligaments are tight. And in this scenario, the lens flattens. So imagine you had something on both sides of an object and you pulled outwards. If the thing in the middle is pliable, it should flatten. And that's what happens to the lens of the eye. When the ligaments pull here, and there would be some on the other side, they pull on the edges of the lens and it flattens. The point of this is to allow you to see distant objects. So when you look at something that's far away, this automatically happens in your eye. If you switch and you're looking at the board and then you go to look at your piece of paper, which I'm sure will happen to many of you multiple times during the day, the other scenario happens on this side. So when they relax, so kind of like a muscle, they don't push and pull, they either pull or relax. But when they relax, the lens is rounded. So if you look at the shape of the lens here versus on the left, it looks a little shorter and fatter than it did when it was being pulled from both sides. Now, without getting too much into the physics of this, if you remember learning about lenses even in junior high science, the shape and size of the lens determines which way light bends. That's basically the principle that we're using here. So when the lens is rounded, it bends the light differently. So here, you can look at close objects more effectively. Now on the bottom, there's just a picture of an eye from the side instead of from the top, showing you how when the lens is stretched, light gets bent at a slightly different angle than when the lens is rounded. Not this specifically. So this accommodation generally decreases with age. So if you have ever noticed that someone who is older than you needs reading glasses, usually the reason is their eye has trouble doing this. Their eye has trouble changing the shape of the lens. The lens gets stiffer and harder as time goes by. So for example, I have good vision because I got LASIK and they fixed it. But I'm starting to notice this happening where if I'm looking at something really close and then I go to look far away, it takes a noticeable amount of time for my eyes to adjust to it. So even though I'm not nearsighted or farsighted, I'm starting to have trouble going from far to close. Not so much that it's affecting my life, but enough that I noticed that it was happening. Yeah, it's the transition. Being nearsighted or being farsighted usually has more to do with the shape of your eye and your cornea. And I don't know if we'll get to that necessarily in detail just yet. We'll probably get to it in our class on Monday, talking about that, but it's a different reason than this. Does anybody else have questions about accommodation? You want to ask? What are we at for time here? Okay. So the next thing I want to show you is a little bit about the organization of your retina. Your retina is cool and kind of weird in the way that it detects a stimulus. Now, I gave you this picture in your study guide not because I anticipate that you are going to memorize the names of all of the cells on this picture. What I wanted to impress on you with this diagram is where the photoreceptors are relative to the other things in your eye. This is the back of your eye, and this would be the front. And you'll notice this is the direction that light is coming from. So when light enters your eye, it goes through several layers of cells. So ganglion cells, amacrine cells, bipolar cells, horizontal cells. None of those are names that you have to be able to recall. I just want you to see there were other types of cells. Until it gets to the photoreceptors right here, the cones and the rods, those are terms that you have to know. So cones and rods are on your retina, which is at the back of your eye, and in the retina, they're at the back of the retina. So our photoreceptors are at the back. This kind of helps explain why you have a blind spot on your eye. Because the photoreceptors are at the back and all of the cells that they're attached to where they could send the signal are in front of them, there needs to be a place where those cells sort of loop around and get out of your eye. So if I think about the signal or the impulse from photoreceptors, so the photoreceptors are here, they are going to send signals outwards. So they'll send signals here, out here, and eventually out there. So the signal from photoreceptors travels to the front of the retina. Now, those cells that are at the front of the retina, so like the inner layer of the retina that's more towards the middle of your eye, those end up making the optic nerve. And you know from the larger picture of the eye that the optic nerve has to come out the back of your eye. So at some point, these cells need to cut through the retina to get to the back of the eye. That's why you have a blind spot. So this signal travels to the front of the retina and then leaves via the optic nerve at the back of the eye. Now, the other thing that you need to understand about how photoreception happens is what kind of stimulus those photoreceptors are actually detecting. So, for them to send this signal or impulse, something needs to stimulate those receptors. And what actually happens is that the receptors, so our photoreceptors, our rod and our cone, uh, are producing an inhibitory neurotransmitter. And they do this unless light is present. So they produce this inhibitory neurotransmitter, but they stop when light is present. So if we could make an analogy of what's happening, it's like there's a brake on, and light being present removes the brake and lets the signal flow. So an inhibitory neurotransmitter, if you remember from the last section, is one that would decrease the chance of there being an action potential in the next cell in the pathway. So if we stop the production of an inhibitory neurotransmitter, we're removing the blockade, removing the brake, and letting a signal pass through. Now, does anyone want to ask any questions about photoreceptors or how the signal is produced or travels? Now, the next thing that you need to know about is visual interpretation. Visual interpretation works like a lot of things do in your body, where things switch sides. What's happening on, say, the left side of your body is interpreted in the right side of your brain. I didn't put this particular diagram because you don't really need to be able to label things. I just wanted to be able to show you that if I'm talking about this side of the brain, it's actually the other eye's information that's being processed. So if we follow the pathway for visual interpretation, we start with the eye, so the photoreceptors, so either a rod or a cone. The signal leaves the eye via the optic nerve. Eventually, it ends up in the thalamus. So you might remember that the thalamus was called a relay station. One of the things it relays is sensory information like vision. And so information gets to the thalamus and eventually to the occipital lobe. But when we're in the occipital lobe, like my picture is trying to show, it's the opposite side from the eye. So most of the information that your right eye sees gets processed in the left side of your brain. Most of the information that your left eye sees is processed on the right side of your brain. Now, it would be reasonable for you to have to identify in which order things happen for you to interpret a visual stimulus. So this is the basic pathway. Sometimes, like if I think ahead to exam questions, sometimes the thalamus doesn't get included. Sometimes we just skip right ahead to the occipital lobe. But certainly this would be the basic pathway. If you look at my picture, it's showing more detail than this, like something called the optic chiasm and the optic tract. You have something called the lateral geniculate nucleus that's part of this. So there's more detail than what I'm showing you, but this would be the basic pathway that things follow. The other thing you need to know about visual interpretation is that we have binocular vision, where bi means two and ocular means eye. So we have two eyes that let us see. Now, there are lots of organisms who have two eyes, but not all organisms have depth perception the way that we do in our eyes. Because both of our eyes are on the front of our head, what I can see with my left eye and what I can see with my right eye overlap with each other. And this overlap is what gives us depth perception. So the fact that you can tell kind of how far or how close something is has a lot to do with the fact that you are going to look at the same object with both of your eyes, get a slightly different bit of information, and when your brain interprets it, it lets you determine how far away the object is. If I think about how depth perception works, it's similar to the way GPS locates you. Usually there's 3 signals in GPS and we're triangulating, but essentially when, say you've lost your cell phone and you're trying to find it and you use the Find My Phone app or whatever app you have, your phone emits a signal, and when at least 3 towers pick up the signal, it's getting the position of your phone from 3 different places, and where they intersect is where your phone should be. When we're talking about binocular vision, you're getting visual stimulus for the same object in both of your eyes, and the overlap helps you figure out depth. Now, does anyone want to ask anything about that idea? If an organism has eyes on both sides of their head, like some fish do, they wouldn't have as much or really any depth perception. They might be able to see things on both sides independently, but depth perception comes from the overlap. Now, the last thing that I want to show you are some eye or vision disorders. And I just want to let you know, in general, when we're talking about disorders, there are so many of them that I couldn't give you an exhaustive list. And usually on a diploma, we're not expecting that you've memorized the details of all of the disorders, but rather, if we described the disorder to you, could you pick out which part of the eye is affected? So, in glaucoma, which is maybe something that you've heard of, there is lots of pressure in the eye because fluid can't drain. Now, if you had to think about the eye and where there is fluid, could anyone think of one of the 2 places in the eye that is full of fluid-like substance? That tear thingy, yeah. So at the front of your eye, you have your aqueous humor. Now, if there's too much pressure in your eye because fluid can't drain like it's supposed to, the result of that would be usually lack of blood flow, which means less oxygen, less nutrients, generally things that cells need to survive. Now the second one is a cataract. A cataract is when the lens in the eye becomes opaque. Opaque is the opposite of transparent. Transparent lets light pass and opaque doesn't. Sometimes the word cloudy gets used here. So in this scenario, because I'm telling you it's the lens, maybe the question could be on a diagram, which part of the eye does a cataract affect? That would be a pretty typical diploma style question. The result of this is that vision is less clear. In other words, acuity decreases. If the lens is opaque and light can't pass through it like it normally should, the retina will not detect as much light or will not detect light in a way that lets you see things clearly and lets you focus. Now the third thing on the list is astigmatism. I had astigmatism in my eye before I got LASIK. Does anyone else know that they have astigmatism in their eye? It's a thing that can make your vision less clear than ideal. In astigmatism, you have an uneven cornea. So if I could just describe what that means, Your cornea is supposed to be this nice, beautifully rounded shape at the front of your eye. But maybe your cornea looks more like this, where it's not perfect, where it's misshapen somehow. Now, if you had an uneven cornea, could someone think of what that might cause for your eye? What might be the result? Stress? Oh, it sure could, but let's just focus just on the light that's coming in. What would happen to light if your cornea was uneven? Yeah. Yeah. So light is not correctly refracted. The cornea is supposed to refract light, and if it's the wrong shape, it's not going to refract light the same way. So I remember my optometrist once telling me that this affected the sharpness of my vision. Not whether I could see something that was close or far, but how sharp it was. So it makes things have blurriness, usually blurry edges, where you can still see the object, but maybe it's not quite as sharp or as clear as you would expect. I can tell you I noticed the difference because when I still wore contact lenses, at first I just had regular plain contact lenses, and then they gave me these lenses called toric lenses. They were weighted so that they would correct my astigmatism, and I noticed a difference, like it was sharper and clearer. Myopia is when a person is nearsighted. If you are nearsighted, that means you can see things that are near, but you can't see distant objects. Nearsightedness in general is something that's hereditary and it's usually dominant. So it would be realistic to predict that in a few years or a few generations, more and more and more people would be nearsighted. When you have nearsightedness, your eye is too long. Now, what does that mean? So your eye is supposed to be a circle, basically. But if your eye is a little bit too long, it's more like an oval. And here's the problem. Your lens is designed to refract light so that it hits your retina. So light comes into your eye and it hits your retina directly because it bends correctly. If light comes in to an eye where a person has nearsightedness, the light still gets bent as if the retina is right here, but it's not. So light is not being refracted correctly, so it's not hitting the retina at the correct spot. Having glasses or contact lenses is meant to correct this. So what the glasses or the contacts do is bend light so that it does actually hit your retina. In a person who has hyperopia, this person is far-sighted. So they can see things that are far, but can't see near objects clearly. Now this could be another reason that someone might need reading glasses. Maybe they suffer from hyperopia. In this scenario, the eye is too short. So the same kind of principle as with myopia, the person has an eye that's the wrong shape, so their lens refracts light at a place that isn't their retina. And then the last one is color blindness, which we've already talked about in terms of genetics. Color blindness is an inability to perceive color differences. And so I already mentioned this earlier, but the part that would be defective would have to be the cones or something to do with the cone's attachment to the optic nerve, but generally the person is missing a cone or one of their cones is defective.